Okay, and was Tara able to join? She was not. Okay. Um, Welcome to the virtual roundtable for the month of May. I think everybody knows me, but we do have some new faces today. My name is Amber Freed, and I am the executive director of Hao-Fountain. Next slide. We have a couple of new faces here. If you would like to introduce yourself, I'll give you a minute. It can just be a short introduction. Okay, well, people are on mute and off video. So if you would like to introduce yourself, just feel free to interrupt or put it in the chat at any time. Slide number 4, Becky, is the beginning of the symposium, and I was gonna have you say a couple of words.
Yeah, sure. So our symposium is going to be in Nashville on September 18th and 19th. Day 1 is going to be scientific. Day 2 is going to be geared towards families. We invite everybody to attend and stay for both days. We're going to have an evening event on the 18th. So we hope that you can join us there. We do also have sponsorships available, and you can find those information about that on our website. You can find information about the entire symposium on our website. Or if you want to just go register, it's usp7.org/register.
Yes, and I'm going to send out a separate email, but we partner with an organization named Uplifting Athletes. And Uplifting Athletes gives travel scholarships to students pursuing their PhD, doing lab work, and the scholarships are really good. I think it's up to $1,400. There are some strings attached. You need to be in the US or Canada, but this is really found money. The other thing that's great about these travel scholarships is that Athletes gives research grants for, and it's later in the year. It is such a fun experience. Everybody receiving an award goes to Philadelphia and participates in a draft. I couldn't remember the word. A draft, just like football, and science is celebrated, but it gives you an into the organization, and hopefully you'll be able to receive a grant later on in the year. So the next slide is of last year, and I cannot say what a great experience it was. Like, all of the scientific learnings aside, we had a fabulous time. Like, I left there feeling so enthusiastic and making a lot of good friends. Next slide. So, just as a reminder, especially to the new faces on the call, we have a full biobank. We have patient samples, iPSCs, whole blood, whatever you need, we got it. And the same with our resource spreadsheet. All mice, the mice phenotypes, an overview of our labs, whatever you need, we really have this. Next slide. We have some new research that showed up in my PubMed feed today with some very recognizable faces within our community. And I will send out a link to this in my my monthly wrap up email. Next slide. We are looking for medical doctors, as I've been talking about, and this has really become thematic throughout the rare disease community. Now, all of our patients historically have been seen by Dr. Christian Schaff, who is now in Germany. So for US patients, it's not as convenient as it was when he was in Houston. And next slide. There are centers of excellence popping up around the US focused on rare diseases. And this is a really dark way of explaining it, but we all know the rare disease statistics, and more people are being impacted, and that includes people with monetary means to make an impact, and also some politicians have been struck by tragedy. And next slide, Becky. Oh, that was out of. Let's go to the next slide. And that includes the Sergey Brin Foundation. Sergey Brin was one of the founders of Google. I actually had a call with them a couple months ago because they wanted to hear what it's like to be a patient organization and how you decide what to fund and what the unmet need is. And their programs were kind of under the radar at the time, and I went into the call knowing nothing because nothing was available on the website. And it turns out they're making $15 million grants to fund what they call the Impact Network, the innovative medicine and precision approaches to clinical trials. And what this means is they want to create a robust and collaborative international network of sites with the goal to accelerate clinical trial readiness and implementation for both syndromic and non-syndromic forms of autism. In Amber Freed terms, they're really trying to make this thought of N of one precision type medicine for monogenic disorders become a reality. And this first phase is for I think about it more as like a hospital-based group of clinicians and researchers to have the ability to enroll and recruit autism patients, no matter what that underlying diagnosis is. And once you create the network of patients to be able to implement clinical trials and show readiness. I'm going to send out more information about this, but I believe the application is due on June 24th. Next slide. Okay, Tara was not able to join, so next slide. Okay, and I was going to introduce our speaker today that I am so excited about, but before we do that, why don't we have Aden introduce himself? And then after he is done with introductions that I just see was put into the chat, we can hand it over to Dr. Allison O'Neill to discuss establishing IPSCs for UPS 7.
Adam just texted in the chat that his
Yeah.
camera and
Okay.
microphone off.
Okay, great. Okay, I will read it.
Okay.
Aden's he says, "Hi everyone. My name is Aden Pinto Fernandez at the University of Oxford. He has been working on dubs, including USP7, for over 10 years. His lab's focus is target and antagonist discovery and characterization. Thanks for inviting me to this meeting. Sorry I cannot use the microphone camera at this time." And there you go, Dr. O'Neill.
Actually, Dr. Besanova is going to go first. She's going to introduce the research first.
Perfect.
So take it
Okay.
away, Irina.
Great. Well, um, can you see my slides?
Yes.
Okay, cool. So I have to apologize. I have to leave at exactly 11 o'clock, just in case we don't finish the discussion. So I would like to do like a small introduction to what Allison is going to talk about, just reminding sort of everybody why we're doing what we're doing with Allison right now. So my lab has been focusing on the impact of Hao-Fountain mutations on USP7 function in vitro, including the mutations we looked at were all within the catalytic domain. And everything that's shown here in blue is something that we've introduced into the catalytic or full-length USP7 one by one. And then my student Emily Korchak, who is on the call today, tested the effect on the protein. This sort of distribution of the mutations that we tested so far, so they're throughout the catalytic domain. And the first thing that Emily did is she tested whether any of these mutations are detrimental to the structure of the protein. Will they just unfold the protein, make it dysfunctional that way? And she found that indeed some were. So we could see for some mutants that they had an effect on protein stability. Specifically, this L2R mutant, L469R, was really detrimental to the protein structure. She also tested the effect on of these mutations on recognition of the substrate, essentially asking the question, is mutation preventing ubiquitin binding, which is something that USP7 does. And again, she saw the entire spectrum. In black, there's a wild type here with just plotting the KD, so essentially affinity to the substrate. And as you can see, some mutants actually made the protein even better binder to ubiquitin, while others completely destroyed its binding to ubiquitin. So like this E345K, for example, completely prevented binding to ubiquitin. And finally, we asked the question, what happens to enzymatic activity of this protein? And then in this case, we're looking at just isolated catalytic domain. And what we expected is to see a loss of function. And indeed, for some mutants, we did see that. So there was a range of mutants here that were less active than the wild type, that's shown in black again. For example, this E345K that prevents binding to ubiquitin also made the enzyme inactive, or this L469R mutation that unfolded the protein and destabilized also made it inactive. So that made a lot of sense. However, we're surprised to find that some mutations actually made the enzyme more active. So we call these hyperactive mutations, like this N418S or G392A or G392D made the enzyme more active than the wild type, at least in the context of the catalytic domain. And there were also mutations that didn't really affect the enzyme activity significantly, so we don't really understand well why just yet. Emily then repeated all of these experiments with the full-length protein, and this is where we saw more differences. So essentially what we saw when we introduced the same mutations into the catalytic domain within the full-length protein, we saw sort of a similar pattern where mutations that disrupted ubiquitin binding or disrupted the structure of the protein were inactive, and other mutations that were hyperactive in the context of the catalytic domain were still active, not more active than the wild type, but still had retained significant activity. So essentially what this led us to believe is that the disease might be caused not only by the loss of function of USP7, but may also be caused by the gain of function of USP7. Then another question that we were working on together with Sarah Borlage at the Dana-Farber Cancer Institute is whether we can rescue activity of USP7. And Sarah's lab found a compound that apparently activated USP7 catalytic domain as well as the full-length protein to some extent. So this is a very exciting finding. We're trying to publish this right now. Emily took that activator and tested it on all the mutations within the full-length protein that we looked at so far. And she found that indeed this activator can rescue some of the mutants. For example, it can definitely increase the activity of the wild type, but it can also increase activity of the many of the mutants, except again for the mutants that have a mutation that's detrimental to structure or substrate recognition. Essentially, those are beyond repair. So this is all nice, but everything so far that we've done was done in vitro. So it's all done on pure protein. So it's great for understanding the mechanism, but it's really not great for moving forward. So what we need next is a cell-based model of USP7 that we can use to test what happens to the activity in cells, not just in the test tube, and also something that we can use to test potential activators and inhibitors of the enzyme. And this is where Allison comes in. So the need for the cell-based model sort of made me look for people that can do stuff like this that my lab cannot. And Allison is in Wesleyan University, so nearby. We met at the conference, so we started talking, and it seemed like a good match. She makes neurons. She's a pro at that. She's a cell biologist and also a biochemist. And we put together a proposal submitted to NIH, a pilot grant called R21 that was designed to essentially understand the effect of these mutations on neuronal development. We picked 2 mutations, and thanks to the combined brain, we're able to get some cells and isogenic controls for one hyperactive mutation and one inactive mutation. Then I guess I'll hand it over to Allison to tell us what she tried to do.
Thank you, Irina. Thank you. So that was a great introduction. I'll share my slides. Can you see my slides? Yes. Okay, great. I will make a laser pointer. Cool. Okay, so thanks for the great introduction. So I'm at Wesleyan University, and my labs uses patient and kind of standard human embryonic stem cells and induced pluripotent stem cells to make the cell type that are affected in neurodegenerative diseases. And so this is our lab's first foray into a neurodevelopmental disease. So I'm really excited to enter this space because there's so much overlap between neurodegeneration phenotypes as well. So just a little background that kind of sets up why we're making the measurements we're making. And so the studies of the actual Hao-Fountain patients showed a decrease of white matter and kind of the smaller brain volume and less complexity of the overall structures. So our question is this from a loss of neurons or neuron complexity? So that would be more of a neurodegeneration phenotype. Or this could also be caused by there not being enough neurons to begin with. So thinking about the neural progenitor pool and how the brain is built from the ground up. And so to kind of reflect on this, USP7 has been shown to be be important for dendritic pruning and synapse density. So I listed the targets here. So one, this XIAP is involved in a tubulin processing. And so they saw that if they knocked down USP7 or activated USP7, so they overexpressed, they could affect how much the neurons arborized, which is a great word because it makes it look more or less like a tree. So it's, are the neurites branched, long, thin? So more branching means more possible connections in the brain. And so healthy brains, neurons are highly branched and also highly dynamic. And then this PPIL4, this is a much more recent study, and they showed that if you decrease USP7 activity, then you get decreased PPIL4, and that decreased the synaptic spine density. And so this is where the synapses are. It's like these little, almost hairs look like structures, these little things along the neurites that are where the two neurons come together so they can talk. And so if you decrease how your neurons can talk, that has a big effect on how your brain works. And so these were all done in mouse. And then we also found some research showing that USP7 is involved in stem cell pluripotency and self-renewal. And so these were done in human stem cells. So the targets of USP7 are NANOG, SOX2, and OCT4, and the deubiquitination of these proteins make them stay around longer, and so helps maintain this pluripotent state. So actually, when we started this, we were wondering if we were going to even be able to take our stem cells and push them out of the pluripotent state, because USP7 helps moderate that. So this is kind of the background. And so we thought, okay, this is these are really measurable phenotypes that we can do in stem cell derived neurons in the lab and on the stem cells themselves as well. So these are the lines that are available through combined brain with their isogenic controls. Isogenic controls mean these point mutations on USP7 have been corrected back to wild type. And so it's these HFS 2, 3, 4, 5, 7. These lines are, we got all of the lines and we banked them in my lab. So we're excited now that we have kind of all of our methods established and troubleshot to go back and try all these other lines. So like Irina said, we started with this G392D and L469R because they're kind of, we thought like kind of opposites, right? The G39D had a hyperactive catalytic domain, while the L469R was a loss of activity. And so remember that Hao-Fountain patients are heterozygous, so they have one copy of wild type. And so we wanted to see kind of how that would rescue if you only had one copy of working USP7, how that would affect the outcomes. We also use this line 1016A. This is my lab's tried and true, amazing iPSC line. This line we differentiate and use right next to these USP7 lines to make sure that the phenotypes that we're getting at the end are because of the mutations and not because of the cell line or because that batch of differentiation didn't go well or something. 1016A differentiates like beautiful. It's just like a beautiful line. If you need a control line, I can't recommend this one enough. It makes great neurons, all types. So the big questions we're asking here are, can we use these patient IPCs to understand the outcomes of patients? So do they reflect do the phenotypes we can measure in the dish, do these reflect what's seen in the patients? And then, thinking about this neurodevelopmental issue, we were wondering if there's a defect in the neural progenitor pool. And so as we differentiate these cells from stem cell to neuron, we go through a neural progenitor stage, just like in human development. And so we're wondering, is there a problem with this kind of building up enough pool so that you could actually build a full brain? And so I'll show you data about the neural progenitors and how we can get neural progenitors, spoiler alert. And then we're asking questions about like, are they actually growing? So at the same rate, because there's this large proliferation stage in development. And so are they growing the same? Do they grow for the same amount of time? And then also just looking at their general health. So looking at DNA breaks, and we could look at maybe some ROS or something to see how healthy they are. The next question is there a defect in the maturation of the cortical neurons? So first question was, can we even make cortical neurons with these lines? If you work with stem cells, you know that not every line, wild type, mutant, whatever, is not guaranteed to work in the differentiation protocols you use in your lab. And that's just something that is just a bummer about stem cells, that not all lines work as equally well. And so then once we make these neurons, we want to measure the neurite length and branching based on what was seen in those mouse models. So the first thing we wanted to do was just make sure that the iPSCs that we got are actually pluripotent. So always good to check. So we did some immunofluorescence for SSEA4, which is a surface marker, and SOX2, which is a transcription factor. So you can see this really nice nuclear staining. So all 4 lines, so the isogenic and mutant, both pairs, are definitely pluripotent. I'll also say I don't have any bright field pictures here, but they have like perfect textbook iPSC morphology. And that was true for all of the lines that we received from combined brain. So we did this as just kind of a check, but I can tell from the bright field that they're all very much pluripotent iPSCs. They have a very distinct look. So just if you're going to use these lines, I would say that they are all truly pluripotent stem lines. So the first thing we wanted to know about these iPSCs is, do they even express USP7? And so, yes, they do. And so here we have that 1016A, our lab's favorite line, and then our isogenic mutant pairs. And so this is just a representative Western blot. And so you can see here's our USP7 band. This is across 2 biological replicates on here. And so you can see everybody has a USP7 band. We normalize this to GAP-DH, and it looks like to me that all of the USP7 lines, they have similar amounts of USP7. So this is interesting because we know this L469R doesn't fold well. So I don't know, but I'm wondering, like, does that mean like there's more wild type or what in this mutant type? Is there some kind of compensation mechanism? The next thing I wanted to know was, is USP7 functional? And so we talked about this at the last roundtable. How do you pick a target of USP7? And so we tried a couple and we came to this P21. And so just to remind everybody, because it seems to confuse people when Hamza, my graduate student, and I talk about it, is that if you knock USP7 down, that means your target's going to go down because it's not going to get deubiquitinated, right? It's going to get degraded. And so if you have more USP7, you should have more of your target. It should be more protected. So we did a functional assay where we took the stem cells and we treated them with this XL177A, a really potent inhibitor of USP7. And so I have those circled. We didn't do it in all lines yet. And you can see that P21 is gone when we use the inhibitor from these 2 different lines. And P21 is highly, really nicely expressed in all the cell lines as well. And so we have 2 replicates of this. And so there are differences. There seems to be some kind of trend that the mutant has more. This needs to be repeated several more times to see if that is true. So if there is more P21, you would think there's more USP7. And so this L469R mutant, remember, knocks down activity. So you would expect there to be less P21 when comparing it to the wild type. So this is interesting here. And we just need to repeat this several more times and see if it's true. So I just wanted to go back and just say, what is directed differentiation? I'm not sure if I have other stem cell people on the call. So we use this new developed rapid cortical organoid protocol. And they call it a cortical organoid. It's a directed differentiation. So what that means is we use small molecules to force transcription factors on and off and turn on and off pathways to push the stem cells through. It's truly a quick walk through human development, which is why I prefer using these kinds of differentiation protocols rather than some of these where you express like NGN2 and you just kind of go right from stem cell to neuron. You miss all this information of the development in between. So even for our neurodegeneration studies and Alzheimer's, I think you should go through this process to better understand what's going on. So with this protocol, you start with stem cells, and then we aggregate them into 3D. So this is different, I think, than many other kinds of cell culture. So we do all of the differentiation in 3 dimensions in a mixing plate. So this is just a rocker like you do your Western blots on, and we have these in the incubator. And so in a 6-well plate, and we have these little chunks of cells swirling around. So the first thing you do is you actually turn off endo and mesoderm, and then the cells turn to default to ectoderm. So we go through this nice ectodermal neuroepithelium kind of expansion. So then the next thing, then you end up with neural progenitors. And so this is that space that I was talking about, is this kind of rapid proliferation step that's seen in the brain and in our protocol. What does that look like between the mutant and the wild type lines? And so then from there, then you do some specification to get to the cortical lineage rather than like the motor neuron lineage, as an example. And then we add factors at the end that help mature the neurons to, and this is the magic part that makes it go faster, just help mature the neurons and kind of seal their fate as cortical forebrain. So let's start with here. So the first question is, can we even make neural progenitors? So this is early days. I don't have a ton of great pictures. I have some good pictures. So here's the isogenic and mutant lines. You can see here's our host. We've got PAX6 and SOX2 expression. These are classic neural progenitor markers. Remember, SOX2 is also a stem marker as well. So it's important that these cells are expressing SOX2 and PAX6. And we see this robustly across all of our cell types. And here's the 1016A kind of trial line. So we were really encouraged because, as I mentioned, we were worried we weren't even going to be able to get these stem cells to stop being pluripotent. So with this protocol, we can get here to the neural progenitors. This is at day 21. We should be able to see them earlier. And I think now that we've been doing some troubleshooting, we'll be able to measure them at earlier time points as well. So then, okay, we have neural progenitors. Do we have USP7? Is it present and functional? Because we're kind of forging at least I haven't seen any published papers using the iPSC lines. We just don't even know if USP7 is there or not. So we've been doing these kind of very basic asking these basic questions. So USP7 is present. We have an N of 1 for all of for 3 of the lines. And so what's really interesting is at this neural progenitor stage, our USP7 goes from a single band. We get this very bright double band. I'm very interested to hear if anybody else sees this and what they think it is. We don't know what this is all about. So we're going to, of course, repeat this. And so USP7, though, is expressed in these lines at the neural progenitor stage. Great. And then we wanted to know, okay, is the P21 there as well? It is. We're going to go back and use the inhibitor and just see if it's the USP7 P21 kind of activity, if that's also intact at this developmental stage as well. And so we have P21. Again, it's pretty similar across the lines, and USP7 is also pretty similar across the lines. So great. Then the big thing is, can we make cortical neurons? To make the pictures big so we can actually see them, I split the 2 pairs. So here's the G39-2D. This is the catalytic hyperactive mutant. TBR1, SAPB2, this was done in the same image, and then CTIP is in a different, well, we can't put all of the antibodies together. TBR1, SAPB2, and CTIP2 are all transcription factors for different layers of prefrontal cortex. And so we were excited to see that we do see TBR1 and we do see SAPB2. If you've got a good eye, you'll notice that there's a lot of overlap of the TBR1 and SAPB2. So that means these are more immature neurons, which is not unusual for us. This isn't a phenotype. And then what I do want to point out is this MAP2 here at the end. So MAP2 is a microtubule associated protein. And so this lights up the neurites, and something that was maybe immediately obvious to me, and I'm excited to see if this is truly a phenotype that after we repeat this a few more times. But Can you guys see in the isogenic, so the top one, there's kind of these nice long, thin, maybe you could see a branch or two, but there's these kind of long spaghetti string-like neurites. Whereas in the mutant, you can't find as many long ones, and they're actually quite fat. They're kind of short and stumpy. And so I'm wondering if this is going to be one of the phenotypes we'll be able to measure. So we do neurite tracing in my lab. So we're going to trace all the neurites, and then we get a per cell neurite length and how many branches per neurite. But we've been successful in using this to characterize neuron health for neurodevelopmental or neurodegeneration diseases. So we're excited to apply that here. So we're excited to see if this is true or not. Got to do it a couple more times. For the L469R, we were also able to make cortical neurons. Again, this is early days, so I had to pick the picture. What pictures we have aren't great, but we do see TBR1 and SAPI2 and CTIP. The TBR1 antibody got left in the fridge for 3 months, so it's staining the whole cell. We see this in our Alzheimer's cells too, all of a sudden too. But what's nice is you can kind of see the neurites here in this green. You shouldn't be able to, but you can because this antibody got screwed up. And this one is the loss of function. And I see some longer, thinner neurites here. So I see some nice long ones. There's also some kind of short, stubby ones. There also seems to be a lot of cell bodies that stain with MAP2 that don't have neurites. So again, we're going to have to quantify all of this. The one MAP2 staining of the isogenic line, that it came out okay. You can see this little bit of neurons, and you can see kind of these long, thin processes. So again, I'm wondering if this is a phenotype, stubby, short neurites or cell bodies without any neurites. So then, are the cortical neurons, does it express USP7? Again, we had to ask this because expression levels change when you go to different cell types. And we do see USP7 across all 4 lines. Interestingly, this double band that was so strong in the neural progenitor stage, we still see it a little bit in this G39-2D mutant, and you can see it in the 1016A a little bit too. This is our lab wild type control. So again, we need to repeat this, make sure our protease inhibitors are all good, and see if this repeats. But I'm interested to see what this double band is. And then do we have P21? We do. So P21 is also expressed in these cortical neurons. And so is this real, these different levels of expression? Because here now we're seeing some differences between, some bigger differences, I would say, in the level when we quantify it. This is N of 1, so we obviously need to repeat it. But we're excited that USP7 is expressed in these neurons, and this target that seems to be working well for us with the inhibitor is also present in this cell type. So we'll see what the inhibitor studies say, if there's the link between USP7 and P21 is still there. So that's where we are. So I feel like we've established with a lot of troubleshooting with the new lines that the USP7 patient lines are a viable model because we can make the cell type that's associated with the disease, or at least the cell type we think is associated with the disease. For all my other neuro people out there, USP7 was shown to not be expressed in glia. So we make astrocytes in my lab on a regular basis. So we could look at astrocytes, but because that was in mouse, but I think that's way down the road just to see what about in these human iPSC derived glia. So next we're just going to, now that we've there's a lot of troubleshooting to get all these protocols up and running for new lines. It always is. And so now we feel confident that we can make neurons, and we're making the neural progenitors. So now it's time to really characterize them. So that's where we are. We're pretty excited. We have our model established, and happy to answer any questions or start the discussion.
Thanks, Allison. I think it's super exciting actually to get somewhere, to get the cells, to get the neurons, and just to see if USP7 is expressed in these mutant cell lines, I think is really, really important because nobody really looked at it yet. systematically. So I think it's a really big step. Thank you. Another question, I know we discussed it a little bit, but I wonder if you want to talk about this, whether we can differentiate between the mutant and the wild type of what's expressing.
What do you mean what it's expressing?
If we express the wild type, if the wild type is overexpressing uncompensating the mutant, or is it the mutant, how much of which is expressed? I don't know if you guys have any insight on how to test this best.
Boris, did you like push your hand up? I don't know, or you just you wanted to make a comment? Does anybody have that that double band of USB 7 on their Western blog? Anybody ever see that before?
I do, but I don't work on like primary cells. I just work in cell lines, and I have like two big band and a thin one as well. I don't know what it is. When I silence using sRNA's USB 7, the two bands are gone, basically. I haven't looked at yet isoform splicing variant, for example. That's something maybe worth looking as well. But,
Boris, you do mass spec, right?
yeah, so that's the kind of thing is tempting to we can cut the band in the SDS page, for example, and run into mass spec. That's an option, but usually it's kind of disappointing because like mass spec is not like protein sequencing. You just rely on like, is the peptide reliable? Can you see anything? So we can try to identify the trisoform. We can cut in the gel. I can ask my Jeff, my PI, who is more like familiar with like the amount of protein you need for that, but you need like large amounts, and we always expect like to see like to identify the peptide from that, but sometimes it's kind of difficult. So yeah, that's a possibility. I wonder as well if you were able to check the localization of USB 7 in differentiated neurons, or is it just expected to be everywhere anyway?
I don't know. Hamza is my PhD student who's been doing the IF. Have you done any IF USB 7 on the neurons? I don't think so.
No, we haven't done any IF. We tried it once with the same antibody that we were using for the Western blot, but it's used in really high concentration. It's not viable with it's not compatible with the IF. So at the moment, no, we haven't checked the localization, but we might. I don't know if you find a good antibody for ESP7.
What is your antibody? Is it the cell signaling one,
Yeah,
or
it's the cell signaling 48SS that's
okay,
used for the Western blots.
yeah, I probably use the same one anyway.
Do you use that one for IF too, Boris?
I did once, but it's true that we did a lot. I didn't have great signal, but it was also like mainly in the nucleus and like some dots in the cytoplasm. I don't know if you expect like a big change when it's a bit everywhere anyways. But, yeah, I guess
We could try it. We could try it, and it might be worth trying actually, and just we can put it on the confocal. So we have a high content fluorescent imager in my lab where we do all these kind of high throughput assays. So, but we could take it to the confocal. That would make a dim signal show up.
Yeah, it's always tricky because if they are like well expressed, like, maybe some people see the ambiguous localization. What?
It could, yeah. Yeah, what do you use it at like 1 to 100 or something? Hamza? Is that what the dilution is? I never trust
I
anyone.
used it at 1 to 50, but I didn't get
I don't
anything.
trust anyone. Yeah. You do that one.
Okay, super interesting. Thank you very much.
I think we got a double band in mice hypothalamus. Somebody in the chat said that.
Oh, I'm seeing in the chat.
Yeah.
In the mice hypothalamus also. Yeah, I wonder if it's a neuron specific isoform. It's interesting that it shows up in the progenitors and then we don't see it in the cortical neurons in all the lines, but we have to repeat this a couple of times and see. See how it turns out.
Awesome. Aiden had a question. Very cool data. You could use activity-based probes to assess activity of the mutants.
What kind of activity-based probes?
Yeah. Well, he doesn't have a microphone, so he has to type it.
I'm going to put my email in the chat, Aiden, and you.
Yeah.
I'd be interested to hear what kind of activity-based probes
Okay.
you mean? Because that'd be great.
Yeah.
That would be really great. So what's nice about using these iPSCs is that we can scale up our differentiations, and we're good at cryopreserving the neurons. So we can make a big batch of neurons, cryopreserve them, and then we can do a bunch of experiments. You don't have to like, okay, today's the day we made neurons, we got to do all the experiments. Like you can freeze them back, and so we can, be a little bit more, plan things out a little bit better. So we can do a study that informs the next study and just pull another vial of neurons out of the liquid nitrogen tank.
Perfect.
Well, I have a quick question out of curiosity. Thank you so much for the presentation. I was wondering if you also checked your experiments in the later stages of maturation, like day 50 plus, just to see, yeah.
Yeah, we have. So Hamza's been trying to keep them, has been keeping them later, and the longer you keep them, the more the organoids get tangled together. You can think about it as like a tangled ball of yarn. So the longer you keep them, the harder it is to dissociate them. So I don't know how successful you've been, Hamza, at getting them apart. We had some day 50 data. Was that?
Yeah, we have day 50 data, and it's somewhat similar to what we have seen already. We have that for the protocol control that we're using, that is 1016A cell line, and for both the mutant and the isogenic. And we have seen like it's not significantly different. It's the same almost. Yeah.
Yeah, I don't remember that all of a sudden they made these big, beautiful neural webs. So we plate the cells after 35 days, 45, we dissociate them from 3D, plate them in 2D, give them 3 days to like heal and put back out neurites. And so by 3 days, you should have a really nice kind of spider web almost. So I would actually say the isogenic lines aren't that great either, which has been difficult. Makes this a little difficult, and that's just stem cell annoyingness.
Yeah, because I faced a similar thing. Either it was so hard to dissociate them or they would collapse. So yeah, I was just curious to see if you saw the same pattern. And yeah, sometimes it's very hard to keep them more than like 50, 60 days or performed experiments on those stages. I mean, I'm not working on the same line, but I was a different line. So
But the iPSCs, I just haven't been able to talk to anybody using the USB7 iPSC lines. So
yeah.
But which line are you using?
It's a GLUT1 line. It was for a side project I was working in, but yeah.
Yeah.
Well, yeah.
We've been working with Dash Y, who developed the protocol that we're using. He's the director of like the neuron program and the stem cell core at Children's Hospital and Harvard Medical School. So a very, very good resource if you ever need help with any iPSC stuff and neuron stuff. But he gave us some tips for doing these dissociations, and he said, don't be afraid and just leave in the accutase for like a half an hour, like, don't be afraid, they can stand it. So we're going to try that because the manual kind of disruption is what kills them, right? And so you just try to be so gentle, but
yeah. And even cryopreservation, I had problem with that. Like they didn't survive after that. Like at least my organoids didn't survive.
So there's a really important thing. You have to freeze them back at 20 million cells per mil, which sounds silly, but it's true. My friend did like a bunch of different concentrations, and like 20 million per mil, you have to be there, and you have to freeze them back in a mil, not a half a mil at the same density. That doesn't work. It's something black magic. And then CryoStore 10 is what works the best. We did a big survey of all the different cryopreservation medias. CryoStore 10 is the magic one.
Yeah, so, good to know. Yeah.
Yeah.
Thank you.
Dash also told us about a new, like, better than rock inhibitor mixture. It's called CEPT, CEPT. So, and he says it's been working like miracles on their stuff. So we bought some. We're excited to see how that goes. It should help the dissociation anyway. I'll say this then, if we can get this scaled up now that we've kind of gotten over some of the bumps in the road, we would have vials of neurons that we could send people as well. So just put that resource out there.
Thank you for saying it. I was like too shy to ask.
So differentiation. So each vial of cells on a good day costs about $125 to produce them if you think about the batch, and that's a good day. So it's stupid expensive to make the neurons. So if you need some, plan out your experiments well. Don't you dare throw any away. Use them all.
And is it like hard to cultivate when you have them? Like, is it hard to maintain? You need like a special cocktail, or is it like everyone can do it?
Yeah, everybody can do it if you can play. If you're using primary neural lines, it's not any harder than that. You thaw them and just you have to plate them on the right thing and feed them the right media, but they're not going to grow. So what you get is what you get.
Yeah, exactly.
That's the hard part.
Yeah. Thanks.
But we can also cryopreserve the neural progenitors as well. So then those are another resource if people were just interested in the neural progenitors.
And then you have to differentiate them in the lab, right?
Yep. Yeah. And you could differentiate. So you can actually expand, make your own bank. And then you can, every time you want to differentiate, you pull a vial of progenitors.
Okay, great.
And so those are growing. So those you can expand and,
Well, listen,
grow up
this is
batch.
so amazing. Like from the PAG side of things, we get questions all the time that is like, this is hard to do, or we find someone who's not keyed into the community and trying to grow something. And this entire presentation is essentially a cheat sheet to success. Like, you've done all of the hard work. So I think I'm going to put this in our patient presentation and just direct people to this conversation. And hopefully we'll be at a point that they can just get neurons directly.
Yeah, that would be an amazing. Again, they're very expensive to make, so I can't just like hand them out like candy.
But we can sponsor that. Like, that is such an incredible use of time to just have neurons made and ready and write a check. Or if it's, I have no idea the logistics that would make it easiest for you, but if we made like a little treasure chest of them and put them with combined brain or something, like, I don't know, I'm just thinking out loud, but that is something we don't even have to think about. Like, let's bathe in neurons and make these widely accessible.
Yeah, I think that could be a really nice resource. So you can also there's several different stem cell cores where you can say, here are these cells I'm interested in, make me neurons, and they can make all different cell types for you and make a big bank. So that probably would, if we wanted to make a big bank to send to combined brain and have just like ready, then you can contract. That's what the cores like are set up to do, right? Like you pay them to do something and they do it for you. So and I would highly recommend Dash's core at Children's. Just he's maintains really high standards.
Well, we are a sure thing. Like, yes, let's absolutely keep this conversation going, and we'll rely on you for cues of when and how to do things.
Yeah, no problem. I'm happy to be a resource for that. And if there's other cell types that people are interested in, that's the beauty of stem cells, right? If people are interested in muscle, we could make muscle. People are interested in dopaminergic neurons, we can make those too. So that's why I love stem cells. Whatever you're interested in, we can make it. We can make gut. I mean, you can do anything.
Well, it looks like we're finishing up on time. Does anybody have anything else they would like to add?
Amber, do you know anybody else who's using the stem cell lines? I would love to compare notes with them.
You know, I need to think about it, but I also think that we've been thinking a lot internally about some open conversations to have at the symposium
Mhm.
that y- just more brainstorming sessions and intellectual banter. And this is a perfect example of something we should discuss with all of the minds in one room. So I'm actually taking notes to leave this as open discussion, and combined brain will be there.
Mhm.
And they know of people we probably don't know of.
Yeah. Yeah.
And maybe it even makes sense. Like one point you just brought up, you can make gut. Well, gut is a big deal in USP 7. And
Mm.
I'm sure it's a case of if you build it, they will come. Like if we put out there, like, hey, we have this. Clementina is, or Dennis is representing Clementina on the call today. She is at jury duty, but she's very interested in gut. And it would be amazing if we could just hand these to her.
Mhm. Yeah. Yeah, there's incredible gut organoid protocols now that are, you get like the villi. I mean, it's like amazing. I have not used them because I'm a neuron person, but the protocols are out there. And
And this is more at like a federal FDA level, but and I know the news is crazy right now, but there have been calls to end, and sorry for the background noise. There have been calls to end animal testing, and I can't see that happening anytime soon. But let's just say in the dream that we're able to, well, organoids would be the next viable option in my non-scientific opinion. And if we could be a front runner on that, like that would be great for a simple repurposed drug to say, look, proof of concept, organoid, let's try.
yeah, yeah, yeah. We can definitely, I think if the preliminary data goes well, because we have these nice, Irina presented activator and inhibitor. So we can see if we can rescue these phenotypes too. So if we can see that in our simpler neuron models, we can definitely go to something more advanced, like an actual intact organoid, and see about that too. But we also do single cell electrophysiology, so I'm interested to do that as well, to see if, how the action potentials of these neurons look like too. That'll be really interesting in the future. I need an army of students.
Well, thank you so much for the presentation. I'm so excited to continue this conversation.
Thank you.
Thank you. Thank you, everybody, for coming.
Thank you. Thank you.
And my email is in the chat. If anybody wants to talk offline, please email me. Thank you.